Good evening from the Forbidden Dispensary. Tonight we're opening a drawer we don't usually open on camera, not because it's dangerous, though it is, but because it refuses to behave. Inside is the slow, strange history of how human beings turned leaves and clay, prayers and stubborn experiment into the medicines and industries that now pretend they were always inevitable. From temple brews carried in chipped bowls to apothecary jars sealed with wax, and from there into glass vials, blister packs, and the bright humming corridors of multinational factories. Forget the tidy white coats and the triumphant headlines that make discovery look like a spotlight and a single face. The real story is dirtier. It smells like smoke-logged workshops. It reeks of accidental poisons and close enough substitutions. It's full of cunning apothecaries, persuasive charlatans, and those small, hard-won discoveries that never look like destiny when they happen. Just a person trying something because the last thing didn't work, and the patient is not getting any quieter. And the truth is this: if you were dropped into an apothecary shop in antiquity, or into a crowded 19th century drug works where the air itself tasted faintly of solvent, your very first night would be a trial. You would learn quickly that medicine has never been a single thing. It has always been craft, commerce, ritual, and risk sharing the same bench. If this is the kind of late-night descent you enjoy, make yourself known. Leave your location and the time where you are. The Forbidden Dispensary likes to see how far our little circle of listeners reaches. Now dim the lights. Let the shelves fall into shadow. And step with me into the corridor where the oldest remedies still whisper. Long before any guild charter, before any printed list, before Latin or Arabic had a neat word for what we would later call a pharmacist, human beings pressed leaves between their fingers and watched what happened. They watched fevers break after a bitter tea that tasted like punishment. They watched wounds close after a paste, mud, and ash, and something green held in place with cloth that had already lived too many lives. They watched children finally sleep after a boiled root that smelled of smoke and rain and the inside of the earth. The earliest pharmacopoeias were not books. They were bodies of experience. They were spoken by firelight, traded at campfires, repeated at hearths. They traveled in the breath of mothers and the memory of shamans. They didn't come with footnotes. They came with scars and with the kind of certainty that forms when you've watched the same plant save one life and ruin another. Archaeologists, patient people with brushes and stubbornness of their own, find traces of that memory in the strangest places. In the matted fibers of a 5,000-year-old basket abandoned near a Neolithic hearth where residue still clings like a thought. In a soot-darkened skull that bears the mark of surgery and, more tellingly, an herb packed into a tooth socket, an early attempt to coax pain into silence. In the cold corpse of Ötzi, the Iceman, carried out of ice from around 3000 BCE, whose belongings included the hard, dry bodies of fungi that modern science recognizes as antifungal and antibacterial. These are small proofs, but the dispensary respects small proofs. They are how big systems begin. Long before writing made knowledge look permanent, humans had already organized a crude pharmacy. Bark and root, fungus and clay, honey for wounds, and wine not only for courage, but as solvent and preservative. Trial and error was slow and sometimes cruel. When it worked, the recipe became valuable. When it failed, the failure often became ritual, because ritual is what humans build when they can't afford to admit they don't know. In Mesopotamia, that instinct observe, test, repeat met administration and script. The clay tablets of Sumer and Babylon preserve not only hymns and laws, but therapeutic recipes and diagnostic lists. The Code of Hammurabi, carved around 1754 BCE, does more than hand out criminal justice. It treats the physician as an official with responsibilities and punishments. Those lines are not romantic, they are managerial. They record a society that expected standards and was willing to enforce them with a severity that made mistakes expensive. Babylonian medicine produced a temperament that feels almost modern in its division of labor. There was the Asu, the practical physician who mixed drugs and performed operations when needed. And there was the Ashipu, the ritual specialist who recited incantations and invoked gods against disease. They worked together because the line between medicine and priestcraft was thin, and because the patient rarely cared why they were suffering, only whether someone could stop it. Tablets like the Diagnostic Handbook associated with Esagil Khan Apli, a scholar priest working in the 2nd millennium BCE, with versions we possess often placed much later, catalogue symptoms, prognoses, and therapeutic approaches with a hunger for correlation. If you see this sign, expect that outcome. If the fever breaks on this day, the patient may live. If not, begin to prepare the family. The desire is familiar. The means are archaic. The stakes are timeless. And the materials, always the materials, came from fields and caravans. Poppy and opium. Resins like myrrh and frankincense from southern Arabia. Copper compounds, woolly roots and hard seeds. Logistics mattered even then, before anyone called it logistics. Clay jars were marked with the names of ingredients. Caravan routes were mapped in administrative archives. Temple storerooms kept registries recording what entered, what left, and what was owed. Those are the industrial hints hiding in what we like to imagine as a domestic scene. Even the earliest pharmacy, if you look closely, is already a supply chain. Egypt, with its slow Nile seasons and temple economies, gave the world a different kind of intelligence. Preservation. Papyrus scrolls from Thebes and Memphis survive like long, thin ghosts. The Edwin Smith papyrus, surgical, copied around 1600 BCE, but likely reflecting older tradition, reads like a field manual, meticulous, practical, unromantic. The Ebers papyrus, compiled around 1550 BCE, is something else entirely. Rambling, almost encyclopedic, a cabinet of hundreds of prescriptions, magical formulas, and materia medica that includes plant, animal, and mineral substances. In those lines, you find honey for wounds, sticky, soothing, antiseptic in practice, and comforting in metaphor. Ointments mixed with fats and plant resins. Lead compounds used with a steadiness that modern toxicology would flinch at. Natron and salts used not only for mummification, but for their drying and preserving properties in medicine. Priests served as custodians of this knowledge. Some, like Imhotep of the 3rd dynasty, were later deified as patrons of healing, as if the culture itself admitted that medicine always carried an aura of the sacred, even when it was simply a person doing their best with what was available. Temple sanctuaries functioned as libraries and pharmacies. They cultivated gardens. They kept pigeons and beehives. They stored flasks of aromatic oil for anointing and for extracting the essences of plants. Trade up and down the Nile and across the Red Sea supplied exotic aromatics and spices that doubled as medicine and luxury. Because in every era, the line between remedy and status symbol has been thin enough to cut. Further east, the great civilizations of the Yellow River and the Ganges developed comparable systems, underpinned by philosophical frameworks that made the body intelligible. In China, the figure of Shennong, the divine farmer, lives somewhere between myth and history. The pharmacological text that bears his name, the Shennong Bencao Jing, reached recognizable form in the Han period, roughly the first 2 centuries CE, though its oral and folk antecedents are older. It lists hundreds of herbs and their properties, giving names and tastes and indications, bitter and cooling, pungent and warming, an entire sensory grammar that would later develop into theories of yin and yang and the 5 phases. The Huangdi Neijing, the Yellow Emperor's inner canon, assembled in forms between the 3rd century BCE and the 2nd century CE, places remedies within a cosmological map. Blood and qi, meridians, seasons, diet, rhythm. From our later industrial vantage, what matters is not whether the map matches modern anatomy, but that early Chinese pharmacology systematized dosage and method decoctions, powders, plasters, and relied on gardens, itinerant herbalists, and state-supported pharmacies in imperial capitals to distribute standard preparations. Certain plants appear again and again, ephedra among them, its stimulating quality understood and harnessed long before a modern chemist learned to isolate an alkaloid and give it a crisp Latin name. On the Indian subcontinent, a similar marriage of observation and tradition gave us the Ayurvedic Samhitas. The Karaka Samhita and Sushruta Samhita, assembled and redacted across centuries from perhaps the late 1st millennium BCE into the early centuries CE, with commentaries thereafter, are not merely lists of herbs. They are manuals of clinical practice and teaching, surgical instruction, materia medica, and a philosophy of health that places digestion, mental balance, and metabolic humors at the center of therapy. Sushruta's chapters on rhinoplasty and surgical technique incisions, sutures, instruments, measure and advanced surgical culture, the kind that makes later historians uncomfortable when they want to pretend progress moves in a straight line. Karaka's sections on pharmacology classified drugs according to taste, potency, and post-digestive effect, prescribing combinations and purifying procedures that often involved complex logistics, gathering roots at specific lunar phases, processing plant materials with oils and decoctions, calcining and purifying metals and minerals into basmas used therapeutically. Trade brought ginger, pepper, and aromatic resins into markets and medicines. Pilgrimage routes and river networks moved seeds and knowledge. Healers, vaidya, seer, midwife were social positions, apprenticeship-based, embedded in temple and court economies. Across these cultures, a pattern emerges. The accumulation of remedies, what we might call materia medica, was not solitary genius. It was communal enterprise economies and rituals intertwined. Shamans and priests wrote incantations. Herbalists recorded empirical effects. Midwives passed along practical recipes. Merchants trafficked in raw substances. Early pharmacopoeias inscribed on clay, written on papyrus, sung by storytellers on riverbanks, were as much inventories and logistics manuals as they were prescriptions. Jars were labeled. Storerooms were audited. Some remedies had to be dried, macerated, dissolved in wine, preserved against heat and time, and this required knowledge of storage. Which resins melted, which seeds spoiled, which concoctions held potency in winter and wilted in summer. The sensory world of ancient pharmacy is infinite in detail. The bitter tang of willow, the resinous smoke of incense, the iron edge of copper salts, the sugar sweetness of boiled grape must used as a vehicle, the soft, almost medicinal sweetness of honey that was both preservative and therapy. And there was darkness too, always. Poisons mistaken for cures, arsenical ointment applied too freely, mercury used with hopeful confidence, the fragile balance empirical observation, ritual protection, community memory is how practical pharmacology became stable enough to be written down at all. And then the Greeks did a strange, decisive thing. They asked for causes. It's easy to mock that sentence from the comfort of modern charts and labs, but the question itself, why this? Why now? Why this remedy and not that one was a lever, and levers move worlds. The Mediterranean had always been a crossroads. Egyptian practices, Phoenician trade, Mesopotamian lists, Anatolian tricks of metal and resin, everything moved by ship and donkey long before it moved by manuscript. But in places like Kos and in the Aegean's long shadow, inventories were reworked into systems. Hippocrates of Kos, around 463 BCE, and the broader Hippocratic corpus did not invent medicine out of nothing. What they did was take a messy mass of remedies and insist on principles. Airs, waters, places could shape disease. Treatments should fit into regimens, not always into potions, into life. Humoral theory, older than Hippocrates, but elaborated by Greek thinkers into a systematic physiology proposed that black bile, yellow bile, phlegm, and blood needed to be in balance for health to persist. The treatment logic was elegantly mercantile in its simplicity. If heat, cool; if dryness, moisten. Dietetics became a pharmaceutical strategy. The prescription could be a table plan as much as a tincture. Change the food, change the baths, open a vein, purge the bowels, move the body, restrain the appetite. The physician's authority grew out of this rationalized regimen. The Hippocratic model cast the healer not as a magician, but as an expert in predictability. Observe signs, set out a plan, test the outcome. That's the beginning of something that will later harden into modern clinical thinking measurement as a kind of moral claim. Not always right, not always kind, but deliberate. Rome adopted and adapted Greek practice, but Mediterranean pharmacology becomes a craft that reaches across space and time in the hands of a particular set of writers-men, whose books would become the spine of later medicine even when their errors became tradition. Pedanius Dioscorides, a Greek physician in Roman service, probably moving with campaigns in the mid-1st century CE, wrote De Materia Medica around the year 60 CE. It is the textbook that 1,000 later compilers would return to, like a pilgrim returning to the same shrine with different questions. Dioscorides described some 600 plants and hundreds of substances derived from animals and minerals, noting appearance, habitat, preparation, uses. Importantly, and this matters to the Forbidden Dispensary, his work was practical. An army surgeon on campaign needed to know which root would staunch a wound, which resin would soothe a dressing, which preparation slowed rot. This was medicine shaped by marching orders and supply wagons. De Materia Medica spread in manuscripts across the Roman world and, with layers of commentary and translation, lasted as the backbone of European pharmacology for about 15 centuries. If you wanted to buy a remedy in Alexandria, Antioch, or Rome, a list derived from Dioscorides or from Galen was often the reference. And then there is Galen of Pergamon, 129 to 216 CE, who took Greek theory and built it into a professional architecture that would be nearly unassailable. Trained in Pergamon's Asclepieion, surgeon to gladiators, later physician in Rome, Galen dissected animals, wrote volumes of commentaries and treatises, and codified pharmaceutical formulation into something like a repeatable production practice. His compound preparations, galenicals, were made from substances beaten, boiled, sometimes distilled, mixed into pills, plasters, syrups. He attached recipes to systems of measurement, to utensils, to a lexicon of compounding that would survive into medieval scripts. Mortars and pestles, scales for weighing botanicals, alembics for separating volatile oils, large-scale distillation refined later, recipes with proportions and steps. A craft with a grammar, a bench with rules. Galen's authority wasn't merely scientific, it was social. He served emperors. Scribes preserved his work. Later monastic copyists treated his words like bones of a saint. Meanwhile, in marketplaces across the Roman Empire, vendors sold potions and elixirs by the cup, while military hospitals maintained chests of standard medicines, and merchant ships carried amphorae of resin and wine that doubled as pharmacological vehicles. So the Roman and Greek legacy isn't just a theory of disease. It's a logistical template, notebooks of recipes, measures, trained physicians, and the beginnings of a supply chain. Armies demanded durable preparations. Merchants demanded items that could be stored and sold. Traders brought cinnamon and cassia from beyond Arabia, frankincense from southern Arabia, myrrh from distant coasts. Long-distance goods acquired medical value as they entered pharmacopoeias. Cities became nodes of supply. Alexandria, half library, half workshop blended empirical tradition with gardens where plants were grown for testing and extraction. Medical schools at Kos, Alexandria, and Pergamon trained apprentices who became the hands of the craft. Then Rome's political structure fragmented, and much of this learned apparatus survived in the least expected places. Western Europe's transition into what we call the Middle Ages saw a contraction of urban networks. But monasteries became custodians of a different economy, book economies. The Benedictine rule of the 6th century, with its insistence that hospitality and care for the sick were Christian duties, meant monasteries ran infirmaries and maintained gardens. The scriptorium copied texts. The monastic kitchen doubled as a pharmacy. Herbs were cultivated in cloister gardens. Ointments were prepared in infirmary rooms. And the abbot storehouses logged medicinal stocks the way they logged grain and wine. Cassiodorus, c. 485–5585 CE, urged preservation of learning as part of Christian duty, and monasteries such as Monte Cassino became repositories for classical texts whose survival depended on the labor of monks. Irish monasteries, remote though they were, became ambulances of literacy, sending manuscript copies of medical texts across the North Sea to the continent and preserving fragments of Dioscorides and Galen. But if the monasteries were careful conservators, the Islamic world became something else, an engine. From the 8th century onward, a translation movement centered in Baghdad's House of Wisdom under the Abbasid caliphs translated Greek, Syriac, and Sanskrit works into Arabic. Hunayn ibn Ishaq, 809–873 CE, and his circle translated Galen and Dioscorides with precision and, crucially, often improved botanical identifications and corrected errors. These translations weren't passive copying. They were active engagement. Scholars such as Al-Razi, Rhazes 854–925 CE, critiqued Galen where observation led elsewhere and produced vast compilations of their own. Al-Razi's Al-Hawi mixes case histories with remedies and experiments. It reads like a clinical mind turned into pages, messy, intelligent, stubborn. Ibn Sina Avicenna, 980–1037 CE, synthesized Greek, Persian, and Indian knowledge in the Canon of Medicine, Al-Qanun fil Tib, completed around 1025. It became a standard in East and West for centuries. Avicenna's emphasis on systematic classification of drugs, diseases, clinical signs made the pharmacopoeia not just a list, but a taxonomy. And then the Islamic tradition did something that would change the physical nature of medicine. It got ingeniously chemical. The name Jabir ibn Hayyan, often Latinized as Geber, hovers over a corpus of chemical experimentation, attributions contested, the influence undeniable. Distillation, sublimation, crystallization, extraction. These techniques altered what a remedy could be. Distilling plant essences produced volatile oils of extraordinary potency. Sugar, introduced and cultivated through trade and agriculture, enabled syrups that preserved extracts and made bitter drugs tolerable. Mineral compounds, sometimes poisonous, expanded the arsenal. Al-Zahrawi, Abu al-Qasim, 936–1013, practicing in Al-Andalus, compiled surgical texts and detailed instruments. Ibn al-Baytar, d. 1248, collected and described over 1,000 simples and their uses. This wasn't just scholarship, it was production knowledge. Islamic hospitals by Maristans in Baghdad, Cairo, Cordoba, Damascus contained pharmacies as integral departments. Pharmacists operated within legal and administrative frameworks. In many cities, they were inspected and licensed, a practical heartbeat of quality control and public regulation. Apothecary shops existed in bazaars. Preparation of medicines became a city trade, requiring scales, balances, standardized recipes. The network supplying these pharmacies was global in a medieval sense. Spices and aromatics moved along the Red Sea and Indian Ocean. Ginseng and rhubarb moved along the Silk Road. Citrus and sugar cultivation spread westward with the Arabs into Iberia. Physicians corresponded across empires. Manuscripts traveled with merchants and pilgrims. Plant specimens were planted and observed in palace gardens from Baghdad to Cordoba. The later European scholar, who would lazily call this a dark age, missed the light entirely. The pharmacopoeia was being transplanted, refashioned, expanded. All of it mattered for the logistical template of a pharmaceutical industry that would come much later. Constraints drove ingenuity: how to preserve an extract for months in heat, how to standardize a pill in a world without printing, how to authenticate an ingredient in a market full of fraudulent substitutes. These problems produce institutional fixes: hospital pharmacies, licensed apothecaries, weights and measures, encyclopedic compendia that record not only the name of a substance, but its appearance, habitat, preparation, contraindications. When Constantine the African in the 11th century brought Arabic texts into Italy via Monte Cassino, and when Gerard of Cremona, 1114–1187, translated works in Toledo, bridges formed. Salerno and Montpellier became centers where the Arabic-Greek corpus informed practice, where physicians argued and tested and taught. The monasteries and the Islamic hospitals were complementary guilds of knowledge, one preserved, the other critiqued and systematized. And there were market responses, apothecary shops in medieval Italian towns, and later guild systems regulating sale and compounding emerged, because urban populations demanded reliable supply. Early European physic gardens and university gardens at Salerno, and later Padua and Montpellier, became living pharmacopoeias where botanists and physicians observed plants, replicated preparations, taught apprentices. The urban apothecary now had scales and written formularies, often in Latin with vernacular notes for copying. Administration of a remedy dosage, form, pill, electuary syrup, method, maceration, decoction, infusion, duration became as much a bureaucratic decision as a folk decision. This slow bureaucratization is one of the key shifts toward industrial pharmacology, and it's worth pausing. The Forbidden Dispensary insists on texture on the everyday practice in those medieval houses of healing. The apothecary's bench smelled of roasted roots, vinegar, and fermenting must. There were marble mortars worn smooth by centuries of grinding, cedar boxes for aromatic resins, shelves lined with labeled jars, some glass, some ceramic, some cloth-wrapped and painted. There was humor, too, in the trade. A bile-heavy patient sent away with a purge that tasted worse than the advice to avoid certain foods. A merchant complaining his tonic made him dull, only to learn it had been adulterated with something cheaper. Regulation could be protective and absurd, inspectors pronouncing the quality of rosewater with solemnity, guildsmen fighting over the proper grinding of an herb as if it were a religious rite. But through it all, habits of exactitude grew stronger, measuring doses, separating compounds, noting symptoms and outcomes. The craft was becoming more legible to itself, and so the stage was set. From earth-scented empiricism of prehistoric kitchens to clay tablets and papyri, into the cool systems of Greek thought, into Rome's recipe template, through monastic gardens and Islamic bazaars and hospitals. A culture of medicines developed that mixed ritual and reason, commerce and compassion. People moved plants and ideas on the same caravans. Physicians argued in margins. The scale transformed. Family remedies became army issue. Army issue became hospital supply. Methods refined. Institutions rose to regulate and certify. The pharmacopoeia was no longer merely the sum of cures. It was a map of supply, a ledger of storage, a rule book of preparation. It was, in short, an early expression of industry. And if there is a dark irony in this long accumulation, it is that so much of it was both brilliant and imprecise. Brilliant in observation and method, imprecise in toxicity and the madness of some mixtures. Yet the networks provided scaffolding for something larger. By the time medieval Europe reclaimed and translated these works, the world had already been rehearsing the logistics of scale for 1,000 years. The herb beds of a monastery and the shelves of a Baghdad apothecary were the small factories of their day. Procurement, labor, apprenticeship, quality concerns, distribution. Modern pharmaceutical industry did not spring fully formed from a single laboratory. It accreted layer upon layer, empirical rules, ritual protections, academic debates, commercial innovations. Listen closely to the quiet work that made modern medicine possible. A monk copying a fragile Greek manuscript by candlelight, a Persian physician noting an oil that preserves a wound, an Arab distiller adding a new flange to an alembic to make a more potent tincture, a traveling botanist bringing a rhizome from a market in Basra to a garden in Cordoba and watching it for a season. These are small acts of logistics and craft. These are the repeated acts by which a tradition of remedy becomes an industry of cure. So when you imagine the long corridor running from reed pens in Sumer to glass vials in a modern lab, Think not of a single moment of invention, but of steady accretion. Scribes, merchants, physicians, distillers, gardeners, monks and millers, each adding a tile to a floor that would support a future edifice. And then, late medieval and early modern Europe, something happens that changes the feel of the room. The apothecary shop becomes not merely a place of remedies, but a map of the world folded into a cabinet. Step inside. The late medieval and early modern apothecary shop wasn't merely a place where remedies were sold, it was a map of the world folded into a cabinet, a geography lesson rendered in drawers and jars. The room itself was usually narrow, low-roofed, and so organized it felt almost devotional. And the first thing that caught you before rosemary's sharp green bite or wormwood's bitter perfume was order. Wooden drawers with Latin labels. Glass jars ringed with the faint dust of whatever had been handled too often. Brass scales hung from a beam like silent judges. Shelves arranged like a sermon. Dried roots from hedgerows in Kent and Lombardy pressed close to powdered coral. Bottles of aquavit distilled in a nearby alchemist's furnace. Later, handfuls of bark whose bitter promise would change empires. Some shops traced their lineage to monastic infirmaries where Benedictine and Dominican friars had perfected balms and syrups. The kind of recipes that survive because they work often enough, and because the institution that holds them can afford to keep writing them down. Florence's famous Dominican dispensary at Santa Maria Novella began in the early 13th century and carried its fragrances and preparations forward for centuries, healing and prestige braided together like herbs drying in bunches. Other apothecaries grew out of town need. Lepers and laborers, merchants and magistrates, all leaving prescriptions and payments in the same little copper box. The orderly interior wasn't upheld only by habit and pride. It was upheld by institutions as much as by mortar and pestle. Guilds regulated the trade with a firmness that was, in theory, for the patient as much as for the apothecary. In practice, it was quietly bureaucratic. Apprenticeships of fixed length, fines for faulty weights, municipal inspections, oaths to use measured doses, bans on selling poisonous wares. By the later Middle Ages, across Paris, London, Venice, Nuremberg, apothecaries had been folded into urban economic regulation. Guilds served multiple purposes. They were quality control, they were trade unions. They were civic instruments and arm of municipal revenue and legal discipline. The gravity of a measured dram wasn't policed by some enlightened public health bureaucracy. It was policed by city councils worried about riots, and guildmasters worried about their reputations and pensions. In London, standards for physicians were formalized when the Royal College of Physicians was founded by charter in 1518. The apothecaries, long allied with the Grocers' Company, did not gain their own corporate charter until 1617, when the Worshipful Society of Apothecaries of London was incorporated. These milestones look ceremonial on paper, but they decided who could compound and who could not. And in deciding that, they shaped the flow of medicines into body soldiers, sailors, the rural poor people who would never see the inside of a lecture hall. The practicalities of licensing and regulation were often more revealing than the pomp. Municipal ordinances required apothecaries to display their mortaria-heavy carved stone bowls when ordered, to disclose sources of imported drugs, to submit samples of syrups for proper concentration, to record sales in ledgers. Physicians fought to monopolize diagnosis. Apothecaries guarded the craft of preparation jealously. There were legal skirmishes over what an apothecary could recommend without a physician's written order. These fights shaped who counted as a healer, and they shaped how knowledge was allowed to move. Apothecaries were also entrepreneurs, members of trading households who bought from merchants, sold to locals, lent cash, and occasionally lent their own authority to a convalescent neighbor. Apprentices learned not only how to grind rhubarb into powder, but how to read a recipe, how to balance an account, how to speak the Latin that shuttled prescriptions from lecture hall to bedside. This regulated urban workshop, tethered to municipal power, would soon meet a world reshaped by ocean voyages. The 15th and 16th centuries did more to the apothecary drawer than any single physician ever could. They refilled it. The Columbian Exchange, named for the 1492 voyage that loosened continents from one another, didn't only redistribute foods. It refashioned the materia medica. European plants sailed west and returned multiplied in consequence, while New World plants crossed to Europe and Asia with promises sometimes fulfilled, sometimes exaggerated. Tobacco, encountered early and described in the journals of Columbus's men, arrived in Spanish courts as panacea and curiosity in inhaled medicine before it became a global vice. Once braided and sold alongside lavender and rosemary, it became a staple of colonial commerce, a cash crop underwriting voyages that in turn kept apothecary shelves full. More consequential than tobacco were other imports. In the high Andes, indigenous peoples had long known that the bitter bark of certain trees eased fevers. They chewed leaves, brewed decoctions, used powders in ritual and practical contexts. In the 17th century, Europeans learned that cinchona bark dark, heavy, punishingly bitter could tame the fevers of malaria. There's a familiar romance attached to the story. A countess, a miraculous recovery, a noble name traveling with a plant. The tale of the Countess of Cinchon, bringing bark to Europe in the 1630s, carries the perfume of courtly narrative. But the Forbidden Dispensary has learned to watch for the fingerprints behind such stories. Missionaries who observed and recorded, indigenous healers whose expertise was translated into European commodity, merchants who moved packets along colonial chains from Lima to Madrid to Rome. For decades, the bark was called Jesuit's powder or Jesuit bark. Jesuit networks, whatever else history assigns them, were pivotal in making local knowledge legible to Europe's markets. This was one of the first New World botanical medicines to acquire transcontinental renown. Other substances followed into the drawers: sarsaparilla, sassafras, guaiacum, names that sound like spells because they were carried in the mouths of people trying to pronounce what they didn't yet own. Coca leaves were observed among Andean workers, chewed to ease hunger and fatigue at altitude. That knowledge crept into European catalogues and centuries later into laboratories that would isolate stimulation into crystals. Commodities moved in crates on the same galleons that carried silver. The Manila galleon sailing between Acapulco and Manila served as an artery through which plant matter and knowledge flowed among Eurasia and the Americas. Merchants who trafficked in spices trafficked in medicinal barks, gums, oils. Europe's appetite for new remedies created pressure. Colonial administrations, missionary networks, private patrons funded botanical expeditions to find more barks and roots and minerals with therapeutic promise. In other words, merchant fleets and chartered companies began to structure the geography of healing. The Spanish flota, Portuguese carracks, Dutch VOC, British East India Company. These were not only machines of conquest, they were supply chains for the apothecary. The new global chains altered not just what sat in drawers, but what health cost in political economy. Cinchona's later history is the clearest example. For more than a century, Europe depended on bark harvested in Andean forests. Bark was bulky. Its active power varied by tree, by harvest season, by handling, by adulteration. Supply was precarious because the trees grew in restricted ranges. Empires learned that controlling a plant source could be as strategic as controlling a port. In the 19th century, once quinine, the bitter active compound, was isolated, imperial powers moved to secure production beyond South America. Botanical gardens and experimental stations became operational arms of empire. Kew and later colonial gardens in the Dutch East Indies and elsewhere tested transplants, cultivated seedlings, converted precarious forest harvest into plantation crop. The Dutch succeeded in establishing cinchona cultivation in Java, the British in parts of India and Ceylon, the French in other colonies. With success came capability. Troops could be sent into malarial zones with the confidence that quinine would keep administrators and soldiers alive long enough to govern. The science of pharmacy and the science of empire matured in parallel. You can taste that parallel if you know what to look for. It's the same bitter. Amid these changes, the conceptual foundations of medicine were jolted by a man who preferred furnaces to Hippocratic diagrams. Paracelsus Philippus Aureolus, Theophrastus, Bombastus von Hohenheim, born in 1493, wandered Europe, hurled invectives at professors, rewrote medicine's songlines in his own impassioned German. He rejected the reigning Galenic synthesis. To him, the body wasn't primarily a balance of humors, it was chemical. Disease might be understood through chemical intervention. Remedies could be derived from minerals and chemically prepared substances as much as from plants. He offered a new vocabulary, the tria prima of salt, sulfur, mercury. And perhaps most consequentially, he insisted that dose mattered, that the same substance could heal or harm depending on amount. That truth is often paraphrased as, "the dose makes the poison." It laid down an epistemic plank the future pharmaceutical industry would walk on with great seriousness and occasional carelessness. Paracelsus wasn't content with ideas. He was a craftsman of the drug. He favored distilled essences and mineral preparations. He claimed success with chemical simples, concentrated remedies targeting disease more directly, he argued, than balancing humors. Colleagues railed against him, often as much for his rhetoric as for his medicine. Yet the chemistry he championed had quieter allies. Alchemists accumulated know-how, miners' observations, apothecaries' experiments, glassblowers' art that made delicate retorts and alembics, distillation, sublimation, calcination terms that had been alchemical lexicon moved into civic laboratories and apothecary shops. Alcohol distilled as aqua vitae became solvent for extracting plant essences, a medium where volatile oils and bitter compounds dissolved and could be preserved. The alembic's curved neck wasn't magical. It was a practical device for condensing vapors, collecting spirits, separating an essence from pulp. In practiced hands, these techniques made medicines repeatable rather than anecdotal. As the 17th century unfolded, chemical thinking matured into iatrochemistry. Figures like Jan Baptista van Helmont wrote of fermentations and chemical causes at the heart of physiology. Robert Boyle's The Skeptical Chemist, 1661, insisted on experiment, reproducibility, and a corpuscular approach to matter. Johann Rudolf Glauber discovered salts and developed processes, Glauber's salt among them, that moved between medicine and industry. The apothecary's bench became a laboratory bench. Precise balances, graduated glassware, standardized reagents tools that allowed a practitioner to measure and compare with confidence previously denied to folk memory. This did not erase folk knowledge, it pressured it. It demanded that the old recipes justify themselves in new language. Pharmacopoeia's approved collections of recipes grew in number and technical precision. London's Pharmacopoeia of 1618 formalized ingredients and processes for English practitioners. Other cities and nations published their own. These were not merely cookbooks, they were early instruments of quality control. How to purify, when to reject, what measures a batch must pass, a vial of laudanum, distilled waters, a weighed dram of rhubarb, all increasingly bound to measured expectations. Then the 18th and 19th centuries made the chemical dream concrete. A young German pharmacist in the early 19th century, Friedrich Serturner, did something imagined for a long time but rarely achieved. He separated from opium a single crystalline substance with potent narcotic properties. Around 1804, he isolated morphine, named for Morpheus, god of dreams. The procedure looked deceptively simple on the page: extraction with water and alcohol, acid-base manipulation to separate components, crystallization to yield a pure material. But in practice, it was patience and hazard. Testing on animals, on himself, on students, watching pain fall quiet under a measurable dose. Morphine altered medicine because it was weighable. Opium varied by freshness, origin, adulteration. Pure morphine could be dosed with a balance. Analgesia moved forward by grams and milligrams rather than charm and guess. Soon other alkaloids followed. In Paris in the early 1820s, chemists Pelletier and Caventou separated from cinchona bark the alkaloids that would be called quinine and cinchonine. They translated Andean bark's promise into quantifiable substance that could be standardized, assayed, shipped. Caffeine, too, isolated in the early 19th century, turned the familiar lift of coffeehouse stimulation into definable agent. These discoveries weren't merely intellectual satisfaction. They were logistics. Armies and administrations could stock and issue drugs with predictable results. At the same time, chemistry itself changed architecture. Wöhler's synthesis of urea in 1828 cracked vitalism's popular grip by making an organic compound from inorganic precursors. Berzelius formalized chemical notation and atomic weights, giving chemists a common language. Justus von Liebig built laboratory pedagogy in the 1830s and 1840s, training a generation to move between bench and factory with disciplined analysis. The condenser that bears his name is more than glass. It's an emblem of reproducibility. This is where the leap from artisanal extraction to industrial production becomes possible. Not only discovery, but training, tooling, and scale. The 19th century mechanized extraction, steam heat for distillation, solvent recovery systems, continuous presses, chemical works that could take tons of raw opium or cinchona bark and turn them into refined alkaloids. The nascent chemical industry factories, often located near coal, water, markets converted laboratory know-how into repeatable processes. And then, in 1856, an 18-year-old in London stumbled into a color that would quietly announce an era. William Henry Perkin, attempting to synthesize quinine, found instead morphine, a purple dye born of coal tar chemistry. That accidental purple soot proved that chemistry could make consistent products at industrial scale. Dye firms would become drug firms because they learned the hard lessons first: how to control temperature, purify, remove byproducts, keep lots consistent, turn reaction into patent into commodity. Firms that began making dyes drifted into pharmaceuticals. Family apothecaries grew into companies or were absorbed by them. In Germany and Britain, companies like Merck originating as an apothecary in Darmstadt and transforming in the 19th century, and Bayer, founded in 1863, embodied the commercial turn. Factories, distribution networks, advertising, laboratories staffed by professionals whose job was to tame variability. Standards followed ambition. Pharmacopoeias matured from lists into enforceable expectations. The United States Pharmacopoeia, first published in 1820, asserted a young republic's demand for purity and formulation. Assay procedures became routine. The physician's order, once a scribble for a compounded mixture, began to call for measured tinctures and later for tablets stamped with a firm identity. Medicine changed its ontology. No longer simply an herbalist's art, therapeutic intervention became applied chemistry. Single active agents, molecules with sharper potency. And with that potency came the double edge. Morphine brought relief and, for many, dependence. Quinine enabled governance in malarial zones and reshaped ecosystems and local economies into plantations and export flows. Patents and advertising began to turn remedies into branded objects. The rhetoric changed from inherited recipe to proprietary formula. The apothecary's ledger gave way to corporate ledgers. If you stand back and look along the line from the drawer smoothed by a medieval hand, to the Jesuit's packet of bark, to Paracelsus's furnace talk, to Sir Turner's crystals, to factory smoke, the continuity is striking. The story is as much logistical as intellectual. Glassware improves, balances sharpen, solvents refine, but also guilds enforce standards, colonial administrations secure supplies. Botanical gardens breed plants for plantations. Companies build factories to turn botanical mass into crystalline principle. The shift from apothecary to chemist, from complex mixture to purified substance, was not inevitable, but it was cumulative. Each innovation, each charter, each expedition, each laboratory note built the architecture that permitted modern pharmaceutical industry to emerge. And beyond the shop, beyond the smell of rosemary and boiled bark, something larger was being constructed, a system of supply, measurement, and chemistry that would allow a molecule to travel reliably dosed around the world. The Industrial Revolution did not merely move machines. It rearranged how people thought about making things, including medicines long brewed in backroom shops, cold smoke and steam engines, the clatter of looms, the new chemistry hidden in coal tar. Everything conspired to create an industry that could imagine molecules as commodities and laboratories as factories. Germany, in particular, showed how dye trade could be transmuted into pharmaceuticals. Bayer and BASF and the cluster along the Rhine and Main learned industrial synthesis as a craft of repetition. Merck's genealogy, the apothecary turning into industrial house, became a template. Then, as the 19th century leaned into the 20th, a man with dyes on his mind would make the next leap feel almost inevitable. Paul Ehrlich watched dyes cling to tissues and microbes. From that observation, he imagined a magic bullet, a chemical that could seek out a disease agent and spare the host. In 1909, his work produced salvarsan, an arsenical treatment for syphilis. It didn't cure everything. It didn't behave like a miracle. But it demonstrated the new interplay of laboratory science, industrial manufacturing, and distribution, synthesized chemical, carefully dosed, deployed at scale. And the factories learned to mechanize not just reactions, but forms, tablet presses, capsule fillers, granulators, coating machines, thousands of pills per hour where an apothecary once ground a mortarful for a week's sales. The questions of the era weren't only clinical, they were logistical. Can we assure each tablet contains the same milligram? Prevent microbial contamination? Ship fragile serums without spoilage? Aspirin acetyl salicylic acid, marketed by Bayer by 1899, became emblem and commodity. Not only chemical achievement, but organizational. Feedstock, reactors, purification, drying, tableting, marketing networks. The pill itself came to stand for industrial medicine. Standardized, dosed, commodified. In the United States, firms like Pfizer, begun as fine chemicals, Eli Lilly and Johnson & Johnson built infrastructure and sales forces. The modern pharmaceutical firm became a hybrid. Chemical works, research laboratory, legal office, and sales organization. It had to be, because chemistry at scale demands business arrangements at scale. But a deeper scientific reorientation was coming, one that would turn medicine from managing symptoms to fighting invisible invaders, germs. By the time the 19th century gave way to the 20th, the most consequential shift wasn't in a new jar on an apothecary shelf. It was in what people finally accepted as real: invisible organisms with appetites and agendas, small enough to be missed by a careless eye and powerful enough to rearrange empires. Once disease could be fastened to a microbe, medicine could stop negotiating with symptoms and start waging campaigns. There had been prophecy before explanation. Edward Jenner in 1796 took cowpox matter and inoculated an 8-year-old boy to protect him from smallpox. Crude, brilliant, and ethically unthinkable by modern rules, it was nonetheless a pivot. Protection as an intervention. But it was germ theory that made such interventions intelligible, strategic, rather than folkloric. Louis Pasteur in the 1860s and 1870s dismantled spontaneous generation with experiments so elegant they still teach humility. He showed fermentation and spoilage had microbial explanations. His lab moved from pure research into applied public health. Vaccines for chicken cholera and anthrax. And then the drama of rabies in 1885 when he treated young Joseph Meister. Across the Rhine, Robert Koch turned microscopy into detective work. Bacillus anthracis, then Mycobacterium tuberculosis in 1882, fastening specific diseases to specific organisms through what came to be called Koch's postulates. Joseph Lister, seeing Pasteur's revelations as a weapon for surgery, introduced antiseptic technique in 1867, carbolic acid, sterilized dressings, insistence on cleanliness that transformed operating theaters from grim gambling halls into places with predictability. It wasn't romance, it was procedure, which is another way of saying it was industry's language entering flesh. These arrivals didn't merely convince surgeons to wash their hands. They furnished a new tactical map. If microbes cause disease, then vaccines, antitoxins, disinfectants, and targeted chemicals could be developed as weapons. Public health, too, learned to speak logistics. John Snow's cholera work in 1854, pointing at contaminated water in Soho, and the sanitary reforms catalyzed by Edwin Chadwick's reports and London's Great Stink of 1858, made a brutal truth visible. Health is infrastructure. Sewers, clean water, regulated food supplies. These were medicines in masonry and law. As germ theory matured, a public appetite for prevention emerged. Vaccination campaigns, first halting, then national, became organized. England's Vaccination Act of 1853 made smallpox vaccination compulsory for infants, a state reaching into the intimate territory of bodies. The policy generated resistance as well as compliance. That argument, liberty versus duty, trust versus suspicion, will return in every century with a new face. Laboratories and institutes stepped into the breach. The Pasteur Institute, founded in 1887, became a node for research and vaccine distribution. The Rockefeller Institute for Medical Research, established in New York in 1901, and later the Rockefeller Foundation, financed campaigns against hookworm and yellow fever and trained public health workers worldwide. In 1890, Emil von Behring and Shibasaburo Kitasato discovered diphtheria antitoxin, inaugurating serum therapy. Neutralized toxins with antibodies harvested from immunized animals. That practice demanded a new kind of factory. Not a dye works, not a tablet mill, an animal stable married to sterile benches. Horses were immunized, their blood drawn, serum purified, potency tested. An artisanal apothecary could not do this. It required capital, land, veterinary expertise, cold storage, glass, and law. It required a state willing to regulate biology. Governments responded, sometimes only after tragedy. In the United States, contaminated vaccines and antitoxins, including tetanus, linked to antitoxin production pushed Congress to pass the Biologics Control Act of 1902, creating federal oversight, licensing, inspection, early architecture of what would become modern quality control. Later, the Pure Food and Drug Act of 1906 began a longer regulatory journey, a public demand that industrial medicine be accountable, not merely productive. In parallel, antiseptics and disinfectants phenol, carbolic acid, later branded household antiseptics, were manufactured and marketed to hospitals and homes. Sterile surgical dressings mass-produced by companies like Johnson & Johnson became part of the new armamentarium against infection. The purpose of medicine was shifting from palliation toward microbial control, and from that purpose came a new research priority. Rather than merely extracting or modifying natural remedies, chemists and microbiologists began searching systematically for agents that would attack microbes or neutralize their toxins. Ehrlich's magic bullet vision, combined with Pasteur's attenuated vaccines and Behring's serum therapy into a multi-pronged strategy: prevent when possible, neutralize toxins, and if necessary, attack the invader itself. The antibiotic revolution did not arrive like thunder. It arrived in stages, each stage full of the same mixture we keep finding in this cabinet. Careful observation, industrial muscle, and consequences no one can fully control. First came the sulfur drugs chemical solutions born from a dye culture. In the early 1930s, German researchers screened dye compounds for biological activity. At Bayer, Gerhard Domach's work produced Prontosil, a red dye that by 1932 saved infected mice and soon after infected humans. The active moiety, sulfanilamide, turned out to be a simpler molecule synthesizable, scalable. Sulfonamides were the first time the modern world experienced what a systemic antibacterial could do at mass scale: knock back infections that had been quick and cruel. But bacteria adapted. Resistance appeared within years. The Forbidden Dispensary keeps a mental ledger of such ironies. The more confidently humans deploy a weapon, the more urgently biology evolves around it. Then the story turned again in a London hospital where a mold behaved like a warning. In 1928 at St. Mary's Hospital, Alexander Fleming noticed that a contaminant penicillium mold had inhibited staphylococcal growth on a petri dish. He published the observation and named the substance penicillin, but lacked the means and perhaps the institutional backing to purify it and scale it into a medicine. That problem fell to a different kind of team. At Oxford in the late 1930s and early 1940s, Howard Florey, Ernst Boris Chain and their group, including the essential, often less sung, Norman Heatley resurrected Fleming's observation and turned it into method: extraction, purification, controlled trials. In 1941, penicillin's first remarkable clinical uses demonstrated promise and fragility. Supplies were tiny, purification was difficult, yields were low, the medicine behaved like a miracle trapped in a bottleneck. War loosened the bottleneck by force. Britain and the United States recognized penicillin's potential for wounded soldiers and mass infection control. Industrial fermentation was enlisted. Government agencies coordinated research and production. The know-how of firms Pfizer, Merck, Lilly, among others, became decisive. The task wasn't to make penicillin in principle, it was to coax a fungus into producing it in quantities that made sense. Early production relied on shallow surface culture, trays, laborious, limited. Breakthrough came with strain improvement and engineering. A high-yielding mold strain made famous in popular telling as the moldy cantaloupe, found and studied in Peoria, Illinois, produced far more penicillin per liter. Deep tank fermentation, pioneered notably by Pfizer, turned penicillin from a cottage craft into factory output. Aerated stirred tanks, nutrient-rich media like corn steep liquor, systems to manage oxygenation, temperature, foam suppression. Then came the chemical engineering, solvent extraction to recover penicillin from broth, crystallization for purification. This was optimized in months rather than decades because the war did not allow leisurely learning. The U.S. Office of Scientific Research and Development and the War Production Board coordinated raw materials and priorities. Factories achieved yields unimaginable a decade earlier. Penicillin became not only a clinical miracle, but a logistical triumph, a product of fermentation, chemical engineering, and wartime organization. A medicine made possible by tanks and valves as much as by microscopes. Almost immediately, other discoveries arrived in a burst that made the soil itself feel like a pharmacy. Beginning in the late 1930s, Selman Waksman at Rutgers and his team searched soil microorganisms for antibacterial compounds. In 1943, they isolated streptomycin from Streptomyces griseus. Albert Schatz, a graduate student, played a crucial role. Later disputes over credit and royalties would remind everyone that discovery is also contract, ego, and power. Streptomycin proved effective against tuberculosis and many gram-negative infections. It too became a fermentation product manufactured at scale. The soil, once just a source of botanical roots, revealed itself as a mine of microbial metabolites. Pharmaceutical companies invested in screening programs, culturing thousands of isolates, building fermentation and purification plants tailored to each product. The effect on medicine was sweeping and practical. Surgeries once reserved for desperation abdominal operations, complex wound repair, became safer because infection, the old enemy, was now manageable. Maternal infections, neonatal sepsis, bacterial pneumonia, leading causes of death declined where antibiotics were accessible. Hospitals reorganized. Microbiology labs became central. Antibiotic formularies and dosing guidelines emerged. Procurement of drugs became predictable, essential. The industry itself changed under the weight of its own success. The economic rewards of discovering an antibiotic were enormous. Firms expanded research divisions, forged alliances with academic labs, built global sales forces. They learned to manufacture under sterile conditions at unprecedented scale. Standard units of potency were agreed upon, cold chains, packaging systems, distribution channels prioritized. Penicillin wasn't just a drug, it was training for an industrial era of biologically active manufacturing. And with new power came new problems, visible almost immediately. Resistance, already observed with sulfonamides, re-emerged with penicillin. Fleming, conscientious and, in hindsight, painfully prophetic, warned in his 1945 Nobel lecture that improper dosing and misuse would select for resistant organisms. Hospitals became evolutionary arenas. Staphylococci and other bacteria that could survive penicillin appeared, and the clinical community learned the sobering truth. Antibiotics respond to selection pressures the way markets do, predictably, relentlessly. Industry and medicine reacted in two ways. They developed new drug semi-synthetic derivatives, new classes like aminoglycosides, tetracyclines, later cephalosporins, and they began, slowly and inconsistently, to acknowledge stewardship, use with restraint, dose correctly, protect efficacy. Early stewardship was patchy. Economic incentives for broad use outweighed caution. Agriculture embraced antibiotics for growth promotion, widening the ecological arena where resistance could evolve. Still, the mid-20th century closed with an ambivalent portrait. Antibiotics had reordered the therapeutic landscape, and the industrial apparatus supplying them had grown sophisticated, centralized, global. Pharmaceutical firms were no longer just developers of molecules. They were managers of complex biological manufacturing systems, legal portfolios, distribution networks, and marketing machines. Their factories and labs became nodes in international supply networks that, when well maintained, delivered life-saving compounds to millions. Yet every victory planted the seeds of a new problem: resistance, regulatory complexity, ethical puzzles of access and use. The Forbidden Dispensary has learned to treat such seeds as part of the story, not as footnotes. And in the quiet after the sweep of industry and science, you can still sense the smells and sounds that made it possible. The faint scent of coal tar that promised color and later cures. The hum of an agitator keeping a precious mold alive and productive. The sharp antiseptic odor of an operating room. The rustle of a prescription pad that once summoned compounding and now summoned a manufactured tablet. The story is impersonal and intimate at once. People learn to scale chemistry into goods. Institutions learn to scale goods into public health. It is timber and glass, microscopes and steam, small labs and vast warehouses, and the uneasy companionship between invention and consequence. And then, in the late 1950s and early 1960s, a different silence fell over hospital wards and regulatory offices, one not born of victory, but of dread. A pill that seemed harmless changed the shape of newborn hands. Thalidomide. The late 1950s and early 1960s left an almost palpable hush in maternity wards and regulatory corridors, an institutional silence made heavier by a single terrible lesson. A small pill, marketed as gentle, could redraw a newborn's body and redraw public policy with it. Thalidomide was sold from 1957 by the West German firm Grunenthal under names like Contagon. It arrived as a soothing sedative, then as a promise for pregnant women, relief from relentless nausea, a quiet night, a sense of control over a body that had become unfamiliar. It moved quickly into general practitioners' cabinets, into bedside drawers, into the ordinary intimacy where medicines live when people trust them. In the United States, a request to market thalidomide was filed by Richardson Merrill in 1960. But at the FDA, a new reviewer named Frances Oldham Kelsey recently arrived and, unromantic in her method, hesitated. She asked questions. She demanded data Richardson Merrill did not have. She delayed approval. She refused to be soothed by confidence. That refusal, praised later, rewarded by President Kennedy in 1962, kept thalidomide out of the broad American market. Europe, Australia, and other regions did not have that narrow gate. the malformations became visible as a pattern only once the pattern had already taken lives. Physicians began to notice rare limb defects appearing with uncanny frequency. Phocomelia, arms and legs grotesquely shortened, hands attached like punctuation marks. In Sydney, William McBride reported suspicions. In Germany, Widukind Lenz did the same. Their letters clinical, urgent crystallized suspicion into evidence. By 1961, the alarm was loud enough that the drug was withdrawn. The image of tiny malformed limbs, the shock of ordinary parents, the sudden realization that safe had been a marketing word rather than a demonstrated fact, these forces pushed governments into reform. There had been regulation before. The United States, for example, had already reacted to another calamity. The 1937 sulfanilamide elixir disaster, in which diethylene glycol used as a solvent killed more than 100 people. That horror produced the 1938 Food, Drug and Cosmetic Act, requiring evidence of safety before marketing. But safety was not the same as proof of benefit, and there was no uniform ethical architecture for how human beings should be enrolled, informed, and protected when experimental compounds entered clinics. Thalidomide sharpened political appetite into law. In 1962, the U.S. Congress enacted the Kefauver-Harris Amendments, driven by Senator Estes Kefauver and Representative Orrin Harris, and propelled by public outrage. The statute was blunt and consequential. It demanded substantial evidence of efficacy based on adequate and well-controlled investigations. It expanded FDA authority to inspect manufacturing facilities. It tightened control over drug advertising, so claims had to be supported rather than puffed. It subjected many existing drugs to retrospective review. This didn't just move bureaucratic needles, it reframed the regulator's job from gatekeeper of obvious dangers to arbiter of comparative claims. That role required machinery, trials, investigators, data, paperwork. Evidence became a currency that law insisted on, not an optional virtue. Those trial methods had roots older than the statute, but after 1962 they hardened into expectation. The randomized controlled trial, so ordinary, now it's almost invisible, had been demonstrated publicly by the Medical Research Council's streptomycin trial for tuberculosis in 1948 under Austin Bradford Hill. Randomization, control groups, careful measurement, statistical humility translated into protocol. In the 1960s and 1970s, the conceptual architecture that we now casually call Phase 1, Phase 2, Phase 3 coalesced as industry institutionalized a sequence capable of generating the kind of proof Kefauver-Harris required. Small safety studies, then dose and proof of concept, then large confirmatory trials. It is easy to forget how novel it was to demand that a molecule be not merely not harmful, but demonstrably effective by standards laid down in law. Ethics marched beside methodology. The horrors exposed by wartime human experimentation had already produced the Nuremberg Code, insisting on voluntary informed consent and the primacy of human dignity. In 1964, the World Medical Association issued the Declaration of Helsinki, giving physicians international ethical principles for research on human subjects. Informed consent, risk minimization, the line between care and experiment made explicit. In the United States, the National Research Act of 1974 helped institutionalize oversight through institutional review boards, IRBs, for federally funded research. The Belmont Report in 1979 articulated 3 principles that would haunt and guide every protocol: respect for persons, beneficence, justice. They sound clean on paper. In practice, they are arguments held in conference rooms at odd hours. Good practice became procedure. Good clinical practice. Once a patchwork of expectations was unified more formally through the International Conference on Harmonization, later ICH. Born in 1990 as a pragmatic accord between regulators in the US, Europe, and Japan, ICH's E6 GCP guideline, finalized in 1996, translated ethics into operational demands. Written protocols, investigator brochures, documented consent, Adverse event recording, monitoring plans, sponsor responsibilities. Clinical trials became industrial projects in the literal sense. Timelines, centralized coordinating centers, data monitoring committees empowered to stop trials when harm exceeded benefit, the language of safety signals and suspected unexpected serious adverse reactions. The Forbidden Dispensary has always found it revealing that modern morality is often encoded as paperwork. Safety also had to extend beyond approval. Regulators learned, often painfully, that initial trials can miss rare harms that appear only in tens or hundreds of thousands of users. Post-marketing surveillance had to be built. In Britain, the Yellow Card scheme launched in 1964 to collect reports of suspected adverse drug reactions. A small piece of paper, modest as a receipt, became a sentinel system. The World Health Organization developed its International Drug Monitoring Program and later supported the Uppsala Monitoring Center as a hub for signal detection. In the US, the FDA's MedWatch program arrived in the 1990s, formalizing flows of reports and the mechanisms for label changes, boxed warnings, restricted distribution or withdrawal. The architecture that emerged pre-market trials, ethics review, pharmacovigilance networks was both defense and admission. It admitted that science is probabilistic, that randomized trials have limits, and that uncertainty carries moral weight that must be distributed between regulators, sponsors, clinicians, patients. It also admitted something else, quietly. Demanding more proof makes development slower and more expensive. Sponsors built larger programs, staffed regulatory affairs departments, validated manufacturing processes to demonstrate batch consistency, compiled library-like applications stuffed with toxicology, pharmacology, chemistry, manufacturing. The scale became staggering, safety improved, opacity grew. The longer and more expensive the path to market, the more precious exclusivity became as the mechanism to recoup investment. And that economic truth reshaped the next decades. When regulation tightened, industry sharpened its other tools. Patents, long-standing instruments of monopoly and incentive, became the currency of modern drug commerce. Two legal shifts around 1980 helped launch a new era. In 1980, the US Supreme Court in Diamond versus Chakrabarti held that a human-made microorganism could be patented. The ruling opened a door for intellectual property claims over engineered life and signaled an expansive view of what could be owned. That same year, the Bayh-Dole Act transformed the relationship between publicly funded science and private enterprise in the United States. Universities and small companies could patent inventions arising from government-funded research and license them. This didn't create innovation from nothing. It created a pipeline of ownership and ecosystem where venture capital could look at a lab discovery and see property, not just promise. Startups flourished. Genentech, founded in 1976 by Herbert Boyer and Robert Swanson, became a symbol of this translation bench biology into marketable product. By the 1980s and 1990s, the market became hungry. Blockbuster drugs, therapies for common chronic conditions prescribed by millions became mythology and business model. Their logic was relentless. One successful drug prescribed for years could underwrite pipelines, pay dividends, justify development costs. Companies pursued antacids and statins, antidepressants and antihypertensives, and in doing so, reinvented how medicines were sold. Sales forces ballooned. Representatives polite emissaries with samples visited physicians. Sponsored educational events and key opinion leader networks softened the boundary between education and promotion. In the United States, direct-to-consumer advertising for prescription drugs became a cultural feature. A 1997 FDA policy shift allowed broader broadcast ads. By the end of the decade, medicine had a new voiceover. Smiling couples on television, calm narration, and a rapid scroll of side effects turned warning into ritual. Patents created temporal monopolies. Those monopolies, paired with pricing strategies and evergreening new formulations, minor tweaks, combination products extended revenue. The Hatch-Waxman Act of 1984 tried to balance innovation and competition by creating an abbreviated pathway for generics while allowing patent term restoration for regulatory delay. It institutionalized conflict, innovators versus generics, monopoly rents versus affordable access. Internationally, the WTO's TRIPS agreement in 1994 standardized minimum IP protections, protecting investment while complicating access in lower-income settings. Political backlash, most visible in fights over antiretroviral drugs for HIV forced renegotiations between property and health. The moral arithmetic became public. The right to recoup investment versus the right not to die waiting. The corporate landscape consolidated. The 1990s and 2000s produced mergers that built multinational giants. Novartis from Ciba-Geigy and Sandoz, Glaxo Wellcome with SmithKline Beecham, Astra with Zeneca, Pfizer's acquisitions to secure blockbuster dominance, Roche's moves to secure Genentech and biologic strength. These weren't just boardroom dramas. They rearranged supply chains, centralized regulatory submissions, closed plants, reoriented research, and the tension between profit motives and public health never vanished. The blockbuster model sidelined antibiotics, short-course, low-margin drugs, unsuited to endless prescriptions despite society's need. Orphan drug legislation, like the U.S. Orphan Drug Act of 1983, spurred development for rare diseases with exclusivities and incentives, but also created pricing strategies that could provoke outrage. Essential medicines like insulin and notorious episodes like EpiPen pricing forced political scrutiny. Regulators and legislators found themselves balancing encouragement of risky research with the imperative to keep medicines accessible. Then molecular biology reshaped the industry from within. Recombinant DNA practicalized in the 1970s and argued over at Asilomar in 1975 gave companies tools to make proteins deliberately. Genentech's collaborations and IPO symbolized the new era. In 1982, FDA approval of Humulin, human insulin produced in E. coli and marketed by Eli Lilly under license, proved that a protein made by bacteria could be safe, effective medicine. Manufacturing changed character. Tablets and tinctures were no longer the only forms. Factories grew living system cells in stainless steel bioreactors, coaxing them to fold proteins correctly and add sugars that made them work. Cold chains became essential. Pharmacy shelves became climate-sensitive ecosystems. Monoclonal antibodies followed. The hybridoma technique developed by Köhler and Milstein in 1975 made it possible to produce antibodies against a single target. Early therapies were crude. Later generations chimeric, humanized, fully human-transformed oncology, rheumatology, autoimmune care. Trastuzumab's approval in 1998 for HER2-positive breast cancer married diagnostic and drug, test the tumor, then treat with a targeted antibody. Precision medicine began to feel less like a slogan and more like a workflow. But biologics carried their own industrial gravity. Mammalian cell culture, glycosylation control, sterile processing, expensive facilities, strict validation. Cost of building and qualifying those plants amplified the value of a successful product, and in turn, the politics of pricing. Gene therapy flirted with the public imagination and then collided with reality. Early trials promised source-level cures. Then came warnings written in blood. The 1999 death of Jesse Gelsinger in an adenoviral gene therapy trial raised questions about informed consent, conflicts of interest, and risk management. Children treated for X-linked SCID with early retroviral vectors developed leukemia due to insertional mutagenesis, proof that a cure can carry a hidden blade. Regulators responded as they always do, after learning the hard way. Pause, redesign vectors, demand longer follow-up, harden oversight. Over time, safer vectors, AAV among them, improved the picture. Later approvals in the 2010s validated the approach, though sometimes with limited uptake and immense price tags. Cell therapies like CAR-T arrived with genuine cures and astonishing cost. They also arrived with a new economic question. How do you pay for a one-time treatment that may cost more than a lifetime of chronic care? Outcomes-based contracts, annuity payments, new reimbursement models. Systems began inventing payment on the fly. To accommodate biologics and advanced therapies, regulators created new pathways. The US Biologics Price Competition and Innovation Act of 2009 created a route for biosimilars, acknowledging that biologics are not generics and require careful comparability studies. Europe built its own guidance earlier. Post-marketing registries and risk management programs became common for gene and cell therapies. Through all these chapters, the scale of what was attempted grew. Labs became factories, trials became global operations. Startups learned to live in the shadow of multinationals. Public funding seeded basic science. Private capital chased clinical promise. Patents and licensing encouraged risk-taking. Regulation demanded proof and follow-up. Ethics became functional infrastructure consent forms, review minutes, monitoring charters. And still the old tension remained. Access versus exclusivity, innovation versus exploitation, hope versus hype. The Forbidden Dispensary keeps one ear on that tension because it is the sound behind the shelves, the low hum under every glossy press release. And now, as the late 20th century shifted toward the 21st, another force began to reshape the market for medicine so thoroughly that the world's pharmacies started to resemble shipping manifests. Globalization, a paragraph in a trade agreement, and a battle over who gets to live first. Globalization. By the last decades of the 20th century, the market for medicine had been remade so thoroughly that a pharmacy shelf began to resemble a shipping manifest. Molecules didn't just travel, they migrated through contracts. They passed through ports and patent offices, through trade treaties and procurement programs. The instrument at the center of this remaking wasn't a syringe or a pill. It was a paragraph in a trade agreement, dry language with wet consequences. The WTO's TRIPS agreement, coming into force with the World Trade Organization in 1995, gave intellectual property a new and almost juridical gravity across borders. Patents, those invisible fences around molecules, were no longer merely national bargains. They became global expectations. For countries with deep pockets and established pharmaceutical industries, TRIPS promised predictability, exclusivity protected by international rules. For others, especially those whose industries had been built on process ingenuity rather than product ownership, the landscape turned dangerous and, paradoxically, promising. Long before TRIPS tightened the screws, India had done something radical in 1970. It revised patent law to allow patents on processes, but not on products for pharmaceuticals. That limitation became a lifeline. It encouraged a domestic industry to master reverse engineering, redesign synthetic routes, reach the same therapeutic endpoint by different chemical roads. In places like Bombay, Hyderabad, Ahmedabad, firms learned to treat chemistry as an art of constraint. They became fast, ruthless optimizers. This wasn't romantic. It was survival. It fed a global demand for affordable medicines. Meanwhile, the United States had built its own balancing mechanism. The Hatch-Waxman Act of 1984 created the abbreviated New Drug Application Pathway, an express lane for generics, while protecting innovators' data exclusivity for a time. The result, globally, was an uneasy duet. Originators relying on monopoly windows, generics relying on legal and chemical creativity to end those windows as quickly as possible. Then HIV/AIDS exploded into public consciousness in the 1990s, and legal differences became moral ones. In wealthy countries, branded antiretroviral regimens cost tens of thousands of dollars per patient per year. In the countries most ravaged by the epidemic, people died without access, watching the same molecules exist on a different economic planet. The Forbidden Dispensary has never forgotten how quickly innovation can become a synonym for "not for you." In 2001, a moment in Mumbai echoed around the world. Yusuf Hamied of Chipla publicly offered a triple antiretroviral regimen for roughly $350 per patient per year, an arithmetic challenge to a global order. Activists and clinicians amplified the challenge. The Treatment Action Campaign in South Africa under Zaki Ahmad fought in courts and streets. Médecins Sans Frontières pushed treatment into places where policy had insisted it was impossible. Pharma and Partners in Health treated access not as charity, but as obligation. The conflict became explicit in 1998 when 39 multinational pharmaceutical companies sued the South African government over legislation intended to improve access to medicines. The lawsuit read, to many, like legalism soaked in funerals. Under global pressure and public fury, the companies withdrew in 2001. The story of antiretrovirals wasn't merely charity or manufacturing, it was collision: law, trade, diplomacy, conscience. That collision forced the WTO into politics. Delegates met in Doha in November 2001 and produced the Doha Declaration, short, blunt, and historically important, affirming that TRIPS should be interpreted to support members' rights to protect public health. Practical machinery followed. The Paragraph 6 problem haunted negotiators. How can countries without manufacturing capacity import generics produced under compulsory license elsewhere? The WTO's August 2003 decision attempted a fix. The fix was cumbersome proof again that law is more often designed to hold than to bend. But it mattered. It created windows through which countries could glimpse policy instruments for access. While diplomats argued, factories hummed. The late 1990s and 2000s saw large-scale offshoring of active pharmaceutical ingredient production to Asia. India's chemical parks and China's coastal provinces became mills for the world's medicines. The logic was logistics: lower labor costs, deep chemical skill, and often looser environmental enforcement. Contract manufacturing organizations offered full package services: synthesis, formulation, fill finish, regulatory documentation. The savings were real, the risks were real too. In 2008, a cluster of deaths and severe reactions in the United States revealed that batches of heparin, a blood thinner, derived from animal tissues but dependent on complex global processing, had been contaminated by adulterated raw materials sourced through China-linked supply chains. The scandal was small in molecular size, outsized in lesson. Supply chains that look like maps can behave like houses of cards when a single node fails. Buyers realized procurement was no longer merely bureaucratic. It was geopolitical. Offshoring extended beyond chemistry into clinical development. Trials grew expensive in the US and Europe. Patient recruitment slowed. Sponsors began to use treatment-naive populations and lower per-patient costs in parts of Eastern Europe, Latin America, India, and elsewhere. CROs became powerful intermediaries. Speed increased. Ethical controversies followed. In India, scandals around informed consent and trial oversight in the early 2010s triggered public outrage and legal tightening compensation rules for trial injuries, stricter approvals, a temporary slowdown in foreign-sponsored trials. The calculus of cost versus oversight leapt from spreadsheets into headlines. Intellectual property strategies evolved in parallel. Originator companies learned to extend exclusivity by stacking protections: secondary patents on formulations, new indications, delivery devices, data exclusivity, trade secrets around manufacturing, litigation strategies designed to fragment and delay generic challenges. Generics developed counters, regulatory study exemptions, aggressive patent challenges, and in the US, Hatch-Waxman litigation as a routine business. Some settlements became notorious: pay-for-delay agreements where a brand pays a generic challenger to postpone entry. In 2013, the US Supreme Court's FTC versus Actavis decision allowed antitrust scrutiny of such deals, a tightening of the patent industrial complex's belt. Global health financing began to reshape markets like a heavy hand on a scale. PEPFAR, launched in 2003, and the Global Fund, founded in 2002, became major buyers. Major buyers negotiate. Their procurement policies favored WHO pre-qualified generics. The WHO pre-qualification program, launched in 2001, became a gate through which many suppliers passed quality systems as passport. Procurement volumes created economies of scale that nurtured generic industries, especially in India, and made ARVs vastly more accessible. The Medicines Patent Pool, founded in 2010 under UN auspices, attempted another lever. Voluntary licensing originators grant rights to generic manufacturers for certain territories in exchange for royalties. It produced wins and exclusions. Middle-income countries with large disease burdens often found themselves on the wrong side of territory maps. Too rich for access, too poor to pay comfortably. Middle-income countries with domestic capacity tested TRIPS flexibilities. Brazil and Thailand, among others, threatened or issued compulsory licenses to lower prices, drawing diplomatic pressure. Thailand's compulsory licensing for certain HIV drugs in 2006-2007 became a case study in the political cost of health policy. India, after shifting in 2005 to product patent protection to comply with TRIPS timelines, still retained provisions for compulsory licensing. In 2012, India granted a compulsory license to Natco for buyers sorafenib, Nexavar, citing high prices and inadequate availability. The world watched because it crystallized the tension: innovation incentives versus the right to health, monopoly claims versus public need. Generics reshaped markets in both numbers and narratives. Teva consolidated into a global symbol of generic scale. Mylan, Sandoz, and others diversified into complex generics and biosimilars. Some became entangled in controversies about pricing and market practices, reminding everyone that generic is not automatically ethical. Price and power can find each other in any model. Regulators responded by intensifying inspections and compliance demands. Quality and trust became currencies as valuable as price. Companies pursued US FDA approvals and European standards. Failures were punished with import bans, warning letters, reputational damage. The world learned that cheap medicine that cannot be trusted is not cheap. It is a delayed cost. Still, the system remained unequal. Low-income countries depended on donor funding for specific interventions, ARVs, antimalarials, vaccines, while broader essential medicines sometimes lagged. Donors could narrow choices by focusing funds on certain products. Access became patchwork. Some regions had abundant supply, others had scarcity and long waits. The inequity was political theater with real body counts. Then the century offered the most vivid stress test imaginable. COVID-19. In October 2020, India and South Africa proposed a WTO waiver to suspend parts of TRIPS for COVID-related technologies. The idea was blunt. In a pandemic, exclusivity should not be the bottleneck. The proposal ignited global argument about patents, about manufacturing capacity, about what counts as technology transfer rather than charity. COVAX, designed to procure and distribute vaccines to low- and middle-income countries, struggled when wealthy governments pre-purchased early supplies. Calls for voluntary licenses, compulsory licensing, and global tech transfer multiplied. The conversation between Geneva, New Delhi, Pretoria, Washington became a kind of new diplomacy. One conducted in press releases and procurement contracts as much as in conference rooms. And the truth that emerged, uncomfortable but clarifying, was that patents were only part of the story. Manufacturing capacity, specialized equipment, skilled labor, regulatory know-how, quality systems. These were bottlenecks too. For biologics especially, you cannot simply print a vaccine like a document. You need trained technicians and validated processes. You need sterile lines and testing labs. You need the kind of institutional muscle that takes years to build. The pandemic forced a simple equation into view. Access is legal, logistical, and political at once. And if globalization is the story of movement molecules, contracts, rules, then the next era is the story of speed and simulation. Digitalization and computational power begin to shorten discovery timelines in ways that would have seemed like sorcery to the chemists who isolated morphine by candle and patience. The human genome era translated biological ambition into datasets. High-throughput screening and robotics industrialized the search for hits. Then algorithms began to rearrange the search itself. Protein folding predictions, machine learning, in silico screening tools that can eliminate vast regions of chemical space before a single molecule is synthesized. The tempo changes, not the stakes. That is where we go next, deeper into the modern Forbidden Dispensary, where the shelves are not only jars, but servers, where clinical trials become networks, where platforms like mRNA turn a sequence into a product with terrifying speed, where the promise of AI collides with the demand for trust. Speed and simulation. If the globalization chapter is the story of molecules moving through ports and patents, the next chapter is the story of molecules moving through computation, through screens lit at midnight, through servers humming in climate-controlled rooms where the air smells less like rosemary and more like ozone and cold plastic. Digitalization did not replace the old dispensary shelves. It built a second set behind them, invisible cabinets full of sequences, structures, and probabilities. The Human Genome Project's near-mystical completion in the early 2000s didn't hand us cures. It handed us an atlas, and an atlas always creates appetite. Targets multiplied, hypotheses multiplied. And with them, the industrial dream of shortening discovery timelines began to look less like wish and more like engineering. High-throughput screening robotic arms moving plates, automated readers turning fluorescence into numbers, had already industrialized the early hunt. Test thousands, test millions, except that failure is the normal currency. Combinatorial chemistry had packed libraries with variation. But the 21st century layered on a different kind of power, algorithms that could triage chemical space before anyone paid for synthesis. Start with structural biology. For decades, protein folding was a notorious problem. How a chain of amino acids collapses into the shape that decides function, binding, and vulnerability. Experimental structure determination X-ray crystallography, NMR, cryo-EM remained painstaking and expensive. Then in late 2020, DeepMind's AlphaFold stunned a world accustomed to slow progress. It produced predictions that, in many cases, rivaled experimental structures. And then something almost unheard of in corporate research happened. Large-scale release. A database of predicted structures for essentially every protein in the human proteome became available, as if someone had quietly duplicated an entire wing of the world's laboratories and placed it online. For drug designers, that meant targets looked less like fog and more like maps. Binding pockets became clearer. The earliest phase of design shifted from guesswork toward a kind of constrained imagination. Propose, dock, refine, repeat. Not instant cures, but more purposeful routes toward candidates. The second wave was machine learning applied to discovery. AI companies multiplying like spores after rain, promising to do for drug development what automation did for manufacturing. Atomwise Exscientia, benevolent AI, recursion names that sound like startups because they are, built around a claim that the search for therapeutics can be accelerated by better pattern recognition. Some of the early stories sounded like prophecy. Benevolent AI suggested baricitinib as a potential COVID-19 treatment early in 2020 by connecting pathways and targets through a knowledge graph. The idea moved quickly into trials. Exscientia and partners announced AI-designed candidates entering clinical testing. The industry listened, investors listened, and regulators began asking the inevitable question: if you claim a model guided your choice, can you explain the model's judgment? The Forbidden Dispensary keeps a skeptical eye on such claims for a simple reason. Biology is not a polite data set. Most machine-predicted candidates still require synthesis, testing, and the slow humiliation of reality. Cells do not read press releases. Still, even if AI does not eliminate failure, It can change the tempo and the cost of failure. It can eliminate thousands of dead ends early. It can prioritize what humans should spend their scarce laboratory time on. That alone is power. Trials changed too. Not only what was tested, but how. The COVID-19 pandemic accelerated approaches that had been brewing. Decentralized trials, remote monitoring, adaptive protocols, platform trials that test multiple interventions under one umbrella with shared controls. The United Kingdom's recovery trial became a masterclass in pragmatic speed. Launched in March 2020 through the NHS network, it enrolled tens of thousands of hospitalized patients with a simplicity that looked almost old-fashioned. Big numbers, clear outcomes, minimal ornamentation. In June 2020, it delivered a life-saving result. Dexamethasone reduced mortality in severely ill patients. The lesson wasn't only scientific, it was logistical. A health system organized as a network can become a research engine when it chooses to. Platform trials like REMAP-CAP and WHO Solidarity showed similar strengths and weaknesses: flexibility, speed, and an urgent need for harmonized data standards. Regulators who had long preferred rigid, pre-specified plans had to learn to accept adaptive designs without surrendering rigor. They bent cautiously. Digital tools made patients into nodes in networks. Wearables, smartphones, electronic health records, the body producing data as it lived. Real-world evidence became a phrase regulators could not ignore. In the United States, the 21st Century Cures Act encouraged the FDA to consider real-world data in decision-making, and guidance documents proliferated. The embrace was cautious, full of language about validation, bias, and model drift, because an algorithm trained on yesterday's hospital practices can become wrong when hospitals change. The industry learned another lesson. Speed is distributive. Faster discovery can produce more candidates and a higher chance of success, but it can also create an arms race in compute and data access that favors capital-rich actors. When the new tools are expensive, the new cures will tend to be expensive too, unless policy and manufacturing capacity intervene. Then came the clearest demonstration of platform technology: mRNA vaccines. mRNA did not appear overnight. It was the fruit of decades of work. Modified nucleosides that reduced unwanted immune activation, lipid nanoparticles engineered to deliver fragile RNA into cells, manufacturing techniques that could produce clean sequences at scale. The foundational work by scientists like Katalin Kariko and Drew Weissman on nucleoside modifications, and by engineers and companies developing lipid nanoparticle delivery, created the platform. When the SARS-CoV-2 genome sequence was published in January 2020, Moderna and BioNTech could design vaccine constructs in days. Manufacturing and clinical testing followed in weeks. By December 2020, the first mRNA vaccines were authorized and deployed. They felt like miracles because the public had not watched the preceding decades of incremental engineering. In the dispensary's ledger, they were the predictable outcome of platform investment meeting crisis pressure. And immediately, the old bottlenecks returned wearing new clothes. Lipid supply, fill-finish capacity, cold chain distribution, trained personnel, quality systems. The speed of design outpaced the speed of production, and production outpaced distribution in many places, and distribution outpaced equity almost everywhere. Automation kept advancing. Self-driving labs systems where robots run experiments guided by optimization algorithms began to take on repetitive work. Microfluidic platforms could optimize reactions quickly with small volumes. High-content imaging and machine vision turned cell-based assays into vast datasets, enabling phenotypic screening at scale. Synthetic biology companies sped DNA synthesis, organism engineering, and biological manufacturing design. The old bench became part bench, part server rack. Regulation followed, lagging as always. Agencies produced frameworks for software as a medical device, for AI/ML systems that may update over time, for decentralized trial conduct. But these are living documents. written in a state of cautious uncertainty. Regulators carry the memory of thalidomide and every other harm. The public carries the memory of pandemics and unmet need. Between those memories sits policy. So we arrive at a threshold. The future now contains tools that can edit genes, engineer cells, and design molecules with a speed that would have seemed like witchcraft to an apothecary grinding bark in candlelight. And with those tools come questions that no algorithm can answer cleanly. Gene editing moved from improbable to practical in a decade. CRISPR-Cas9 burst into the scientific mainstream in 2012 through work by Jennifer Doudna and Emmanuel Charpentier. And the toolbox expanded base editors, prime editors, delivery systems, and a growing alphabet of engineered variants. The clinical stories already feel like fever dreams from the perspective of earlier medicine. CAR T-cell therapies, approved in 2017, showed that a patient's own immune cells could be engineered and returned as targeted killers, producing remissions in refractory leukemias. Gene therapies offered one-time interventions for rare diseases, sometimes with real restoration of function. And then the price tags arrived like a second diagnosis. One-time curative therapies priced in the millions are defended as necessary to recoup R&D, manufacturing complexity, and small patient populations. Critics argue that prices reflect market capture and what payers can be pushed to tolerate. Payment models, outcomes-based contracts, annuities are being invented on the fly. None are yet fully stable or universal. The danger is obvious. If the next generation of cures scales without a commensurate rethink of finance, disparities will harden into biology. Who can afford to be cured becomes part of who gets to live a normal life. Ethics cuts deeper when germline editing enters view. In 2018, the announcement of gene-edited babies by He Jiankui in China crystallized global alarm and prompted calls for moratoria and stronger governance. The scientific community responded with speed, insisting germline editing was premature and dangerous. But the tool itself is inexpensive, portable, and democratizing. And democratized power includes democratized risk. Global norms without enforcement are only wishes written in polite language. Meanwhile, antimicrobial resistance burned as a slow, steady threat beneath the glamour of gene editors and AI dashboards. The 20th century's success made antibiotics feel permanent. Economic incentives made them disposable. Firms retreated from antibiotic R&D because new antibiotics used sparingly do not generate blockbuster revenue. At the same time, agricultural use and environmental contamination, including documented effluent from manufacturing sites in parts of the world, accelerated selection for resistance. Policy responded with a mix of push and pull incentives, funding programs like CARB-X, industry-backed initiatives like the AMR Action Fund, and experiments with subscription-style payment models that delink revenue from volume. Promising, partial. True stewardship requires coordinated global surveillance, regulation of agricultural use, and environmental controls that treat antibiotic discharge as a public hazard, not a minor compliance nuisance. Environmental sustainability is no longer a footnote. Pharmaceutical manufacturing consumes energy, solvents, water. Discharges alter ecosystems. Biologics production demands cold chains and complex materials. Climate events disrupt supply chains. Investors and regulators now demand environmental metrics and risk mitigation, and the industry is learning slowly that the future includes carbon accounting as surely as it includes potency assays, which leaves policymakers with hard choices that look technical but are moral. Should we extend exclusivity to encourage development of socially valuable drugs like antibiotics, even as exclusivity often raises prices? Should we build public manufacturing backstops for essential medicines, treating supply as infrastructure rather than commodity? Should we fund prize models or patent buyouts, delinking innovation from scarcity? Should we insist that publicly funded science carry public interest licensing terms as default, not exception? We can imagine a pharmaceutical century built on three stubborn pillars. First, distributed manufacturing capacity, regional hubs for vaccines and biologics, technology transfer paired with training and regulatory strengthening, not merely shipments. Second, governance for data and algorithms, shared standards, privacy protections, audit trails, bias monitoring, so AI-assisted decisions can be interrogated and trusted rather than worshipped. Third, realigned incentives for antimicrobials and neglected diseases, subscription models, market entry rewards, sustained push funding, and environmental stewardship enforced with seriousness. None of these is a panacea. The politics of medicine will always be a contest between private profit and public need, between the abstract language of innovation and the immediate breath of a patient. But history offers precedent. ARV access battles showed that law can bend toward life. Pandemics showed that manufacturing alliances can be mobilized with strange speed when the world decides it must. The story of the pharmaceutical industry has never been linear progress. It is a hall of mirrors, science, law, capital, and conscience reflecting and distorting one another. As gene editors refine their tools and AI suggests molecules in the night, as rivers carry the effluent of a century of chemistry and climates rearrange supply routes, our choices will decide whether new treatments echo old injustices or begin to correct them. The ethic we need is simple and stubborn. Discovery fast enough to save lives, governance strong enough to distribute those lives fairly, and one last fact remains, one that no treaty or algorithm can legislate away. Medicine is, at its root, a social act. Labs invent, factories make, regulators approve, merchants distribute. But the test of success is whether medicine arrives where bodies and economies meet. If the coming decades give us targeted cures and price tags that consign them to only the wealthy, the triumph is hollow. If AI shortens discovery but amplifies bias, we've traded one injustice for another. The quiet triumphs are different. A generic pill reaching a rural clinic, a regional vaccine plant staffed by trained technicians, an AI-guided molecule becoming a cheap, effective therapy instead of a luxury object. These are incremental. They are logistical. They are the things a historian later points to and calls evidence. So in the soft late hours of an industry that never truly sleeps, the Forbidden Dispensary allows a kind of optimism, rooted not in wish, but in strategy. Law can be bent toward life, manufacturing can be reconfigured, data can accelerate discovery. The hard memos are not only in lab notebooks, they live in procurement contracts, patent offices, ethics committee minutes, and in the negotiations that convene in Geneva and New Delhi. If the century ahead is to be the pharmaceutical century in anything but name, it will be governed by a quiet equation. Scientific possibility multiplied by political will, divided by the arrogance of letting markets decide alone. And that, in the end, is the oldest lesson in this whole cabinet. The herbs were never enough without the people willing to carry them. The knowledge was never enough without the institutions willing to protect it. The cure was never enough without the decision to share it. Good. Quiet then. Because after all the empires and assays, after all the charters and court cases, after all the petri dishes and patents, the story always ends the same way it began, with a human body in trouble and someone reaching for a substance they hope will help. Here in the Forbidden Dispensary, we keep a shelf for that humility. We keep it next to the shelf for arrogance, because they are neighbors in every century. When you look back along the corridor from the campfire teas and the clay tablets, from temple storerooms to guild ledgers, from Jesuit packets of bark to deep tank fermentation, what you see is not a straight line of progress. You see a braid. Ritual with reason, commerce with compassion, courage with carelessness, each era insisting it is more enlightened than the last, then leaving its own quiet disasters for future regulators to study. We learn to label jars, and then we learn to label harms. We learn to standardize doses, and then we learned that standardization can standardize injustice if access is restricted. We learn to isolate crystals, and then we learned that crystals can become chains, patent chains, supply chain chains, price tag chains. We learned to build factories, and then we learned factories can save millions while spilling their costs into rivers no one wanted to look at. We learned to run trials, and then we learned trials can be both protection and performance-proof and theatre depending on who controls the questions. And now we have algorithms that propose molecules in the dark, and gene editors that can rewrite inheritance, and platforms that can turn a sequence into a vaccine before the public has learned the name of the virus. We can move faster than ever. We can also make the same old mistakes at higher speed. The dispensary does not hate the industry. We do not worship it either. We know what it is: a machine built out of human needs and human incentives. A machine that contains genuine miracles and ordinary paperwork, tender clinicians and ruthless contracts, careful scientists and eager marketers. A machine whose failures tend to land on the people who had the least power in the negotiation. That is why, at the end of this long walk, we return to the bench. Imagine the old apothecary room at dusk, mortar dust floating in a slant of light, labels in Latin and vernacular, vinegar and rosemary in the air. The last customer has gone, the ledger is closed. Outside, the street settles. Now slide forward in time. Imagine the hum of a fermentation tank at night, a factory worker checking gauges, a quality control analyst staring at a chromatogram. The quiet fatigue of somebody whose job is to prevent one bad batch from becoming tomorrow's tragedy. Imagine a trial coordinator answering a late call, a patient signing a consent form with hands that shake a little, a nurse counting pills, a pharmacist watching supply levels drop again. It is all the same work, wearing different clothing. The copying monk and the regulatory reviewer, the caravan merchant and the procurement officer, the herbalist and the computational chemist. Different instruments, same old responsibility. To handle fragile substances with care and to remember that the person at the end of the chain is not a market. If there's a lesson we keep, locked but not hidden, it's this: modern medicine was not born in a single lab, and it will not be saved by a single breakthrough. It is built and rebuilt by systems, supply systems, testing systems, trust systems. Break those, and even the most elegant molecule becomes useless, or worse. And yet, despite everything, we continue. Because people continue to fall ill. Because someone continues to ask the way the Greeks asked. Not only what helps, but why. Because someone continues to grind, measure, test, record. Because even in the most industrial era, the most human desire remains embarrassingly simple: to reduce suffering without creating new suffering as the price. So tonight we close the drawer slowly, not because the story is finished. Stories like this don't finish, but because the hour is late, and the dispensary knows the value of stopping before we start pretending we can control what we're describing. If you're still here with us, wherever you are, whatever time it is in your room, remember, these medicines, old or new, are never only chemistry. They are agreements. They are the shape of a society deciding what it owes its sick. Good night from the Forbidden Dispensary. We'll keep the lights low, we'll keep the ledger open, and we'll be here the next time you want to step back into the cabinet and listen to what the glass and paper have been trying to tell us all along.